So she'll be covering in chapter 4 together with Dr. Nurul. And then we have our pengarah, Dr. Aziah, will be teaching on digital transformation in healthcare together with our fourth invited lecturer from CDEC. It's an electrical engineering center under USM, Dr. Asro. So this is Dr. Asro. So he'll be covering the topic chapter 5, subtopic for digital transformation together with Dr. Aziah. And then the last lecturer for this course is invited lecturer from international. This is Professor Mehmet Ozos. So Professor Mehmet Ozos is our adjunct professor from University of East Side, Turkey. So he'll be teaching all of you on chapter 3, subtopic diagnostic CRISPR, utilizing CRISPR technology. Okay, so these are the teachers. So as a bit of introduction to the course, so basically the course is to share some comprehensive material with students on omics in molecular medicine research, with special focus on omics as a solution for therapeutics, prognostics, and also diagnostics. Beside that, this course also covers some few subtopics that involve disease prevention measures utilizing omics, risk stratification, integration of digital transformation, machine learning, and also artificial intelligence in molecular medicine. Oh, I forgot. Dr. Nadir will not only cover the machine learning, but he also covers the artificial intelligence in molecular medicine. I think this is all together in chapter 5 as well. So this course is divided into 5 chapters, and it is 100% coursework. There's no final exam. So you need to, the assessment of this course will be attendance of lecturers, tutorials, discussion, and also other student-centered learning activities that include quiz, assignment, and presentation. Okay. So the chapters 1 will be current trends and omics for therapeutic solution. Chapter 2 will be current trends and omics for prognostic solutions. Chapter 3, current trends and omics for diagnostic solution. Chapter 4, disease preventive measures and risk stratification utilizing omics. And the last chapter is digital transformation and artificial intelligence in molecular medicine. Okay, so these are the chapters, and these are the course assessments. So there will be altogether 9 assessments will be collected from the students, and it involves assignment, presentation, quiz, tutorials, and also discussion. The assignment, there will be 2 parts in which I myself and also Dr. Venu covering chapter 1 and chapter 2. Okay. And then for chapter 3, there will be presentation by, you need to submit it to Dr. Heng and also Dr. Hairul. And then there will be quiz. So this quiz covering chapter 1 and chapter 2 only. And then there will be tutorial exercise 1, sorry, mistake typo there, and then tutorial exercise 2. So tutorial exercise 1 will be given by Dr. Nurul. Tutorial 2 will be Dr. Shahida. And then last but not least for the discussion, Dr. Atirah and also Dr. Azia. So again, there will be no final examination. So the assessment for this course, the FM 529, will be collected from all this. So you need to submit, compulsory submit all this to the respective lecturers and also do the quiz as well as the attendance lah, so that you can get most marks for the course. Okay. Now, before I move on, space, do you have any question regarding the assessments and assignment or whatnot? Any question? Noah? Okay, before I move on, for the quiz, there will be only one quiz after the chapter 1 and chapter 2. But I'm allowing students to decide when you want to do the quiz. So I would say right after chapter 1, chapter 2 completed. But if you think you need some time to study, then maybe a week after chapter 1, chapter 2, or at least lah, I mean that's the latest I can offer. A week after completing chapter 1, chapter 2. And then for the time, you need to discuss among all of you when you want to do the quiz. It will be an online quiz. And then once you come up with a time, I will set the quiz when to open and when to close. So the quiz will be 1 hour quiz. And the question format is short answer or short essay. Okay, in 1 hour. Okay. Now, if there's no question regarding the if no question regarding the course, introduction, and assessment, whatnot, I will continue with a bit of introduction about Omics. And then I will move on to the Omics as a solution for therapeutics 1. Okay? Now, I'm sure all of you have heard of Omics. And when you think of Omics, probably, I don't know, sometimes we've heard of Omics and we might say, oh, Omics, I know Omics. But when it comes to courses and whatnot, you have to define Omics in a sentence, in a sentence, in a complete sentence. So Omics, it's a study, it's a discipline, it's a field, it's a type of study that cover the roles, relationships, and actions of the various types of molecules that make up the molecular map of diverse signaling and regulation within a cell of an organism. So that's the definition of omics. And this omics field can encompass genomics, proteomics, transcriptomics, and there are more latest omics coming out, like glycomics, lipomics, and metabonomics. I think metabonomics is not that new. It emerged right after genomics, proteomics, and also transcriptomics. So genomics is the study of genes in the whole genome and their function. Proteomics is the study of proteins. Metabonomics, cellular metabolism, transcriptomics, study of the mRNA or transcripts, glycomics, the study of cellular carbohydrates, lipomics, this is the study of cellular lipids. But then now I think probably there are a lot more of new omics. In fact, if you're studying immune system, there is also immunomics, okay, the latest one. Now, the first omics was genomics. And then probably you would ask, what's the difference between genomics and genetics then? So basically, genetics is the study of heredity. And then genetics refers, or gene refers to a specific sequence of DNA on a single chromosome. Genetics involves the study of functions and composition of a single gene. So that's genetics. It's a much small area of study. You're studying a single gene. When the term now changed to genomics, it is a study of the entirety of an organism's genes, and it refers to an organism's entire genetic makeup. It addresses all genes and their interrelationships with each other. So that's the difference between genetics and also genomics. And then the next question you should ask is, how do you generate omics data, or how do you collect omics data, or what is omics data? So omics data is basically a large biological data sets collected from high throughput data collection using tools and technology. So when we say large, it involves probably a few hundreds or thousands of data, and it is collected from a high throughput technologies. And these technologies, if you think of it, the one that very close to, when it close to genomic is NGS is one of it, next gene sequencing. And then for protein, we have spectrometry, mass spectrometry, or maybe HPLC, or gas liquid chromatography and whatnot. So high throughput technologies produce big data. Okay, so these data sets of a biological molecule is called omics data. And then the next term is informatics. Informatics harnesses the power and possibility of digital technology to transform data and information into knowledge that people use every day. This strong focus on the human use of computing helps people to interact with technology in the best and most efficient way possible. So when you have omics data, you cannot get away from analyzing the data using computer science and also, tools, software to analyze the big data, because you can't simply use Excel or GraphPad Prism to do the analysis of big data. You need to use a much more analyzing software. But don't worry, this software, sometimes it's easily accessible, and sometimes it's free. When it say easily accessible, it can be freely used from a public domain database. And unless if you're a computer science engineers, in which you create algorithm and whatnot, generating, what do you call that? Um, what do you call that? Coding, yeah. Generate coding and testing it to create programs and software and tools, whatnot. So that's not our problem. We leave that to computer science people. We just tell them the problem, what is the solution if we want to use to analyze this and not. So we work together closely with computer science engineers to help us to analyze the data and to get the conclusion that we want. Okay? So software. Informatics involve a lot of tools. That tools are softwares. Okay? So historical advances in omics. I think this is just something that I love to share with students. Whenever you studying something, you need to understand the history or at least know the history of it. So I found this in one of the journal. The title of the journal is Genome, Transcriptome, and Proteome, The Rise of Omics Data and Their Integration in Biomedical Sciences. It's quite latest, 2018, so you can check this out lah. So when it comes to data, in fact here you can see the discovery of floppy disk. The discovery of floppy disk is considered as one of the technology or tools for the storage of the big data. So now if we talk about, I don't know whether you guys know floppy disk or not. I'm sure you guys are in the era of thumb drive, but mine, we use floppy disk back then. So, so yeah. So now we have cloud system to store our big data. So we just need to buy what capacity, the size of the cloud system to store everything. Yeah, so it's good to know the history so that you can appreciate more. Huh? So this is to show you the workflow or protocol of omics. So basically omics, you have a sample. Of course, when you are studying a disease in biomedical sciences, you have a model of what disease or what situation, disorder or whatnot that you want to study. It may not be a disease, it may be a physiological understanding of certain process. Okay? So disease is just one of the simplest question that we want to answer. But it may be if you want to understand certain physiological process, that can also be, the model, the question that you want to answer as well. So here, we start off with sample collection, of course, and you need to decide whether it's tissue, blood, urine, and other, etc., etc. So in my case, the model or the question or the problem that I want to answer is breast cancer. Breast cancer. So I have to have a sample, and this sample can be something that I can get from patient if I have connection with the hospital or clinics. So I work together with the clinician, apply ethics, whatnot, and get the sample. I have from patient, and I need to compare it with healthy individual. So I need to decide if it's blood, It's easy. I can get from patient and then I can get from healthy individuals. But when it comes to tissue, I may get tissue from tumor tissue from patient, but how do you get healthy tissue from healthy individual? You don't just simply cut healthy individual. Let's say in the case of breast cancer, you don't simply ask people to cut their normal breast for your research. So that's never happened lah in the real world. So what we do when you have tumor tissue, you collect from the same patient the area that not affected by the tumor. So maybe it's on the same breast, you have the tissue affected with tumor, and then you get the tissue from the unaffected area. So that's the best that you can do lah when it involves tissue collection. Okay. So these are the things that the challenges that you may face when you collect samples. If it's urine, or blood, or follicle hair samples, it's you can compare. You can get from healthy individuals, but when it involves tissue, it's a bit challenging. Okay, because you cannot simply get it from normal person. But what we do in the routine is we get tissue from the same patient, but at the area of unaffected one. Okay, so from that samples, you need to decide what data that you want to generate. Is it genomic, transcript? You want to collect DNA? You want to collect RNA or protein? Or maybe you want to analyze certain metabolite? Okay. So you need to decide, because from there you will choose the technology that you want to use to generate the data. Okay? And then once you have the data, you need to process it. Quality control, normalization, transformation, redundancy, removal, whatnot. I think for this data processing, I think bioinformatics cover it in TFM 522 or something. No, not 522. 522 is diagnostic. The one that Dr. Eugene and Dr. Nick taught you. Okay? Data processing for Omics. Correct me if I'm wrong, yeah? Okay. So anyway, so after that data processing, you have data analysis in which the same data, but you need to do some analysis. And this analysis involves statistical analysis, enrichment analysis, biochemical network analysis, network mapping analysis, clustering, and whatnot. So this is under the data analysis. And then here is basically a lot of tools, software that you need to use to do the analysis for you. And here is where you need to work together with computer sciences to answer certain objective that you want to answer. And then last but not least, you have your output. And from this output, you can extract biological meaningful information for clinical relevance. And this output may be used as a diagnostic, therapeutics, or prognostic. So this is basically what this course, TFM 529, is all about. Okay? Okay. So this is the same workflow, but in the data acquisition, you may don't want, or maybe in the sample, you may don't want to collect sample because you don't have clinician working together with you. And also you may don't have technologies like NGS or that special technologies. But you want to assess what this data can provide you to answer diagnostic, therapeutics, and prognostic. So this is where we call you acquire those data from public database, and then you process, and then you analyze it, and then you answer, you get the output that you want. So here, you don't start with sample, and you don't run the sample on the technology. You get the data from the public domain and you do the downstream processing analysis and you got the output. So this is what I mean by that. So secondary data collection is where you use data from others. So other people have done the NGS comparing metastatic breast cancer and compare it with the non-metastatic breast cancer. So you can get all this data in the public domain. So these are example of external public databases, public domain. TCGA, if you're working on microRNA, and then Entrust Gene ID, Assemble, Romin, Unigene, Uniprot, KEBI. So these are external databases where you can get omics data. You can download it from this database and then you can do the process and analysis. Okay, you just need to search what you want to study. For example, you want to compare immune system in breast cancer patient, compare it with normal people. So you can search that in online in this database, what people have done, and then you can download their data, omics data. For example, if it's transcriptomics or genomics, you can download them. Okay? And then you use tools to do the analysis and the process and whatnot. But if you want to generate the omics data yourself, you can start off with sample collection and then you choose your omics and then you choose the technology. So either the hard way or the easier way. Okay? So whether to do the hard way or the easier way, the one thing that I want to remind you is that a lot of research these days are done in the Western world. So many of the data, omics data, are coming from Caucasian, especially from the Americans and also the Europeans. And these data might not represent your question population, for example, Asian population, So you may go to certain databases that contain Asian. Okay? If you don't bother, you don't mind about whether they are Asian or they are Caucasian or whatnot, that's fine. But when it comes to certain condition or disease like cancer, we know that cancer, the factor that contribute to cancer, the risk and whatnot, include ethnicity, include, the lifestyle. So in certain population, they have a different lifestyle, diets and whatnot. So getting secondary data from, public data domain may not be helpful for you to understand. So in that case, you have to go from zero lah, starting from sample collection, generate the data using technologies, and then process it. So there are the easier way you can say I'm using the data as a general population, but when you check this database, of course, they include the information about the ethnicity, the population, the demographic of the population. So these are the questions that you need to ask before you choose which way to go lah for omics. So genomics is basically the initial omics, start off, and then transcriptomics, proteomics. And then after that, we have epigenetics. Genetics become genomics, transcript become transcriptomics, protein become proteomics, epigenetics become epigenomics. Omics behind there is to indicate that there is a big data involved in the analysis. And then these are what we call as vertical omics. And then there is multi-omics. If you study one epigenomics or one proteomics, you choose one only, we call it as vertical omics. And then if you choose multiple level of this, it's called multi-omics. And then again, this is, I mentioned earlier, the emergence of another type of omics, metabolomics, metagenomics, immunomics, glycomics, lipomics, and probably certain people would claim, certain molecule that they are studying, and probably it covers only that. So they put omics at the back of their type of molecule. So it can be a lot more, but this is just a few. There will be a new emerge omics level to study. And when we say this emergence of new omics, and possibly they are available in public domain as well. So if you're interested with omics study, the best way is to continue reading and explore. Okay, so this is nothing. So yeah, so we move on to this one. I just want to share with you one example of study publication involving how studies of omics have helped us to understand the differences between certain condition and certain condition. Okay, so this paper was published in 2022. Multi-omics, when you say multi-omics, means it study different level of, it could be combination of proteomics with transcriptomics and also epigenetics or genomics, I don't know. But for this one, they claim as multi-omics because they study metabolites, metabolomics. They study lipids, lipidomics, and also they study proteins, which is proteomics. So 3 level of omics they studied here. And the situation or problem that they want to understand is COVID-19 compared to bacterial sepsis-induced non-COVID-19. ARDS stands for acute respiratory distress syndrome. So they do comparative study between COVID acute respiratory distress syndrome to bacterial sepsis-induced acute respiratory distress syndrome. And in that comparison, they collect 666, more than 600 of metabolites and also more than 1,000 of lipids and almost 300 of proteins. And then from here, we present a first comprehensive molecular characterization of differences between the two disease, and further investigation into the identified pathway will lead to a better understanding of the pathophysiological process, potentially enabling novel therapeutic intervention. So this is one example, very simple, of how they want to compare these two types of disease using multi-omics approach. Now, so remember this type of paper, and then what you need to understand here is the first one that you need to ask is whether they collect samples and they run the assay themselves using certain technologies. And then all the processes is just informatics pathway and whatnot too, that involves softwares. But the one that differentiate between, are they starting from zero, they collect samples, or they use public domain data? So from this manuscript, I think they don't start with the sample. I think they use already published data and then they process it. But anyway, so when you say you start off this sample and then you need to choose the technology to generate your data. So I think this comes to the very basic in semester one. You've learned, you have been introduced to many technologies, like how to run the technologies, at least the theory of it, NGS, microarray, HPLC, mass spec, whatnot. I think for these tools, I don't want to go one by one for it, because what's important is you need to understand that, you need to analyze the data to answer whether the omics can provide you solution for therapeutics, for diagnostic, or for prognostics. Okay, so again, this is just to have a recap on how you collect data using these technologies. If it's nucleic acid, it can be sequencing, microarray. If it's protein, it can be mass spec, LC, HPLC. But in fact, for microarray, there is also protein microarray, right? So again here, see, I did highlight here protein microarray, but I put under nucleic acids. So these are some of the few technologies to generate big data, the omics data. And it can also be modified form of these technologies as well. So you can search lah from your reading what kind of technologies people have used. So informatic tools, omics data processing and analysis. So these are some of the routine that people have line when you do omics analysis. So I'm not going to go through omics data processing, because I think under that omics data processing, it's already been covered by Dr. Eugene and also Dr. Nick Hughes. I haven't checked what they've covered, but I already confirmed with Dr. Nikyus that under that course, they did not cover the data analysis part for Omics data. So under here, data analysis, you may come across identification of differentially expressed genes. So these are the terms that you will probably identify under data analysis, which is DEGs, differentially expressed genes. And then you have identification of common genes, enrichment analysis of DEGs and also common genes using these particular tools. Example here are the tools. So these are database. GeoExplorer is database. Interactive Venn here is tools. Go Ontology, Kyoto Encyclopedia of Genes and Genomes. These are KEGG for the enrichment analysis. And then you probably will come across the term protein-protein interaction, PPI network. And the tools that you can use to do the analysis for PPI network is STRING. STRING stands for Search Tool for the Retrieval of Interacting Genes and Proteins. And then there is this one, the very famous Cytoscape is another. So once you, from all those analysis tools, it will generate for you this kind of data. I'm sure you have come across this type of data presentation. So this is what we call volcano plot for differential expression genes. Blue for downregulation. Black means no differences, and red means upregulated genes. And for protein-protein interaction networks, you may see some strings of connectors that connect the whole genes that being discovered. And then this is another PPI network. When there is a string connectors like this, immediately it's a PPI, protein-protein interaction networks. And then you have enrichment analysis in which, using the tools, you may identify certain biological process involved as a living beings, whether it's under cellular biological pathway, cellular biology pathway, and whatnot. So this is what we call enrichment analysis. And then Venn diagram is a representative data for you to show that you want to compare what are the genes that common between the two problems. So the red one is the genes for the problem number 1. And then this blue one is gene or any omics for the problem number 2. So Venn diagram will do the merge and see what are the common gene that connect the two problems. So here when you have 2 problems, you will see 2 round here. But if you have 3 problems, you want to compare 3 different situations, you will have 3 round thing here, and then you will find in the middle where it connect with each other. So this is what we call Venn diagram. But of course, this is the first. You need to get the database. You need to get the omics data from the public domain. And I mentioned earlier, there are a few public domains where you can download the the data, and then you just decide what analysis you want to do. Okay, so that's basically the whole thing about this course. Omics data, you want to do the easy way or the difficult way to generate your own, or you want to use public domain data. But at the end of the day, you are going to do the analysis of that data, the DEG, the DEG analysis, protein-protein network enrichment, and also common genes identification using Venn diagrams. All this to answer one, whether it's to search for novel gene for therapeutics, novel gene as diagnostics, novel gene as prognostic. Okay, so this is why we split the chapters into 5. For example, here, current trends in omics for therapeutics. The motivation is to use the omics to predict treatment response and drug development. So this is the motivation for omics for therapeutic solution. And in chapter 2, current trends in omics for prognostic solution, the motivation is omics based to predict risk of severity and recurrence of the disease. So this is the motivation for prognostic. Chapter 3, current trends in omics for diagnostic solution. The motivation is omics-based, including biosensor technology in disease detection, early screening, and disease susceptibility. So these are this is the motivation for chapter 3. Chapter 4, disease preventive measures and risk stratification utilizing omics. The motivation is to use omics to predict side effects and toxicity and to stratify patients, whether the patient is a slow metabolizer, moderate metabolizer, or high metabolizer. Chapter 5 is digital transformation and artificial intelligence in molecular medicine. The motivation is advances in informatics, computer science and information technology, and automation to improve healthcare to manage illnesses. So basically, these are the chapters, and the brown color is the motivation for each chapter. So I hope students are very clear about this. And then if you are clear, and then you would have a better argument when you learn about these certain topics or chapters for this course. Okay. I already shared this with the lecturers, and I hope lecturers also get the same motivation. And when they provide example for you, they provide you the correct example to answer this motivation. Okay. Anyway, so chapter 1. So this is when I start with the chapter 1, current trends in omics for therapeutic solution. So what a long way to introduce this course 5 to 9, because I want the students to really understand what is this course all about. Okay. This motivation that I've listed here is very important for you to understand why we create these chapters. Okay. So The chapter 1, current trends in omics for therapeutic solution, general outcomes to understand the approach and application of omics as therapeutic solution in molecular medicine, to discuss the basis of omics therapeutic solution, to explain the current application of omics as therapeutic solution. So I myself will give you a general basis of this current trends in omics for therapeutic solution. And I may provide you one example. I think the one that I'm providing you will be breast cancer and also another disease. We will reach there. And then Dr. Shikin will cover example of disease, cancer, and again, it's breast cancer, but in her way of interpretation lah, for the motivation, to answer the motivation. Okay? So this is just to recap what is the flow protocol for omics. You have this in mind, and then you will move forward what you want to do. So omics is basically a biomarker. And then for therapeutics, inside the omics contain biomarkers. You screen for these biomarkers inside that omics data, and you enrich, you analyze to find the most useful biomarkers. Let's say you started off with 11,000 omics, and then you come after your analysis, you have scaled down, you identified 20 biomarkers from that 11,000 using all the analysis that you have chosen, softwares. Okay? And then these biomarkers, in the end, you want to use it for what? For this chapter 1, the biomarkers discovered from omics data is to be used as targets with drug to control or combat certain disease. Okay? So this is the motivation for this chapter. Now, when you say druggable targets from omics data, omics-based data, biomarker screening, and then get certain genes as a target biomarker. So basically, for these therapeutics, we call it the very closest term for this is personalized medicine. Personalized medicine is an emerging practice of medicine that uses an individual's genetic profile to guide decision made in regard to the prevention, diagnosis, and treatment of disease. So this is the definition for personalized medicine. Prevention, diagnosis, and treatment of disease. So that's personalized medicine. And then a form of medicine that uses information about person's own genes or proteins to prevent, diagnose, treat disease. In cancer, precision medicine uses specific information about person's tumor to help to make a diagnosis, plan of treatment, and find out how well treatment is working or make a prognosis. Precision medicine has taken precedence for reasons of clarity. The concern with the term personalized medicine is that it implies treatments and prevention strategies that are bespoke to the individual. These connotations are potentially misleading, so precision medicine is currently favored term. So not long time ago, the first term that used was personalized medicine. But now they revise the term as precision medicine. But basically, it's the interchangeable term. It's like when you do real-time PCR, you have housekeeping gene, right? You remember? And then now they change it to reference gene. But basically, it's the same thing, reference gene and housekeeping gene. So the same here, personalized medicine as well as precision medicine. Precision medicine is a more revised version for the term. Okay? Precision medicine, also called personalized medicine, helps helps doctor find a unique disease risk and treatment that will work best for you. Precision health is another broader term that includes precision medicine, but also approach occurs outside the setting of the doctor's office. To protect their health as well as steps that public health can take, sometimes called precision medicine health. But anyway, that's another term, precision health. But I think I don't want to go deeper on these terms, precision health. So we stay under the precision medicine. So basically, what it means by that is, again, you have all these clinical sample to start off, and then you do multi-omics study, you profile the omics of that particular patient, okay, because we are talking about precision medicine here, so I say patient. So you profile everything about that particular patient, proteome, transcriptome, genome, micronomic, metabolomic, and whatnot, all the omics. And then from this omics data, you have certain biomarkers, and these biomarkers are being used to drive drug development. And also there are biomarkers that are being used to drive prediction. Stratification is here. And then from drug development, verification of predicted changes, blah, blah, blah. And then drug development go to clinical trial. And this is where with one patient, you will have one personalized intervention for your particular disease. So this is what it means by personalizing or precisioning medicine for certain patient. Okay, but I'm sure this is far beyond what we can do now, because technology itself is quite expensive, and not all drugs are available to target certain genes. We need to have more and more database of drug that can be used to target all of our genes as human. So a long way to go lah for precision medicine for one patient. But the motivation is there to have a tailored medicine for certain patient with certain omics profile towards, better management of certain disease. Okay. So omics-driven biomarkers, predictive biomarkers to measure the likelihood of response or lack of response of a particular therapy and allow identification of patients most likely to benefit from a given treatment. So thus sparing other patients from toxicities of ineffective therapies. So I'm sure in a semester 1, you have been introduced to what it means by predictive biomarkers, diagnostic biomarkers, prognostic biomarkers. I don't know whether you remember it or not. But under omics-driven biomarkers predictive, this omics data is being used to predict whether patients are most likely to benefit from certain given treatment. Okay, so the one example that the easiest one I can provide to you is in the case of breast cancer. There is several type of breast cancer. You have a hormone positive breast cancer, and then you have the hormone negative breast cancer. For hormone positive breast cancer, there is this particular drug called tamoxifen. So clinical research report have shown that this drug is able to inhibit tumor growth in patients with positive hormone receptors. But in patients lacking the hormone receptors, this drug, tamoxifen, may not benefit them. In fact, it will give rise to toxicities because drug is toxic anyway. So if you don't have that target in your body, why are you taking them anyway, right? So might as well don't take it. Get an alternative drug to counter the disease. So that's what it means by predicting benefit from a given treatment. Okay. To predict drug sensitivity relevant to precision medicine. So this is what it means by um, uh, predictive biomarkers. So companion diagnostic medical device, often an in vitro device which provide information that is essential for the effective use of a corresponding drug or biological product. So in order to predict whether patient is will benefit from certain drug, you will use certain assay, and that assay is called companion diagnostic. So this companion diagnostic will provide you some information. If you are positive for that particular biomarkers, you will have benefit with against that drug to be used with the drug lah. So companion diagnostic assay is one of the, how do I say, one of the thing that is important in precision medicine, especially in cancer. So compare the one that started off with precision medicine is cancer drug in cancer patient. Okay, so that's why you can see here a lot of disease are cancer type: colorectal cancer, non-small cell lung cancer, gastric stromal tumor. Only one here, it's non-cancer, thalassemia. Okay. Okay. So omics-driven therapy decision directly informed treatment in patients and were correlated with better OS, overall survival, and patient PFS stands for, I forgot, patient. So anyway, so basically current assay to help decide whether therapy is beneficial is based on this platform: immunohistochemistry, PCR, MRI, immunohistochemistry. My question is, are these platform omics technologies, high throughput? Well, basically they are not lah. These are low throughput platform. So the current companion diagnostic to assess therapy benefit, they are not omics based because they don't produce high throughput omics data. But the future is omics based companions diagnostic, in which the assay will now start to utilize NGS, mass spectro to assess benefits of certain drug to patient. So most of the omics-driven companion diagnostic now is still at research level. And if it does, definitely not in our part of the world lah. Probably most of them are in the high income countries, the Westerns, and if it's Asian, in the Japan and probably in Korea, but not yet in a much lower middle income class country. Okay. So the future is to provide access to these omics-based companion diagnostics for therapeutics for all, not only for those who are rich. So that is why I said here, is it ready? Well, it is not ready. It's still in a research, and if it's ready, only in the high-income countries. So patient-focused multi-omics information integration can improve the detection and advancement of combination therapies, treatment of diseases with more than one drug, to attain efficacy with smaller doses and a smaller drug toxicity. So these are the advantages of using multi-omics information integration to improve patient management. So this is one example that you can read. I found this and then I've read it and it's quite interesting. The changing landscape of NGS implementation in molecular diagnostic laboratories. So even though probably you might think that, wait a minute, we are talking about therapy here, but how come there is a molecular diagnostic term there? So again, and that's why I said you need to be clear with the term precision medicine for you to decide whether certain drug is working. You need diagnostic assay. Okay, so this is when molecular diagnostic comes in with therapy. But maybe if you read more, you will see the differences between diagnostic. Like I said here, if I go back to the motivation, Chapter 1, omics-based predicting treatment response and drug development. That's the motivation. If you want to predict treatment response, you still have to go back to the diagnostic, create an assay diagnostic. And that diagnostic assay is called companion diagnostic. And the omics era, that assay need to be omics based. Okay? In the chapter 3, omics for diagnostic solution, the motivation is for disease detection, early screening, and disease susceptibility. Disease susceptibility meaning that you need to run omics-based assay to assess whether patient has a higher risk of developing that disease. And usually this is diagnostic assay to detect mutation, certain variant mutant. Okay, so remember this one slide is very important for you to understand the motivation of each chapter so that you won't be confused. Dr. Fahma's chapter 1 is on therapeutic, but why Dr. Fahma include diagnostic here? Because the motivation is to predict treatment response. And that prediction involves precision medicine and involves companion diagnostics to assess the level of expression of certain biomarkers. Okay? Anyway, so omics. So other than predicting treatment response, treatment benefits, omics also can be used here to drive drug development, to design and develop new drug, or maybe to use the biomarkers identified from the omics analysis and to use it under the drug repurposing, using what we already have to uncover new purpose of the drug. Okay? So when I say that, this is an example of one study my student are currently working on. Kirsty. So Kirsty is my PhD student. So this is her project, identification of metastatic breast cancer and tolerogenic dendritic cells. So she needs to identify hub genes between the two conditions, metastatic breast cancer and tolerogenic dendritic cells. And then the hub gene that she identifies, she will use it to discover drugs available already used in clinics and to repurpose it, to repurpose the drug already in clinic. Let's say the drug is being used for cardiac disease, but now she wants to from her analysis, she wants to repurpose it for breast cancer. Okay. And also, this is one particular anti-cancer drug from this particular plant lah, because we have it in our lab, so we want to study it. So this is the omics level that Kirsty using is transcriptomics. It's a vertical omics. And also it's immunomics, sorry. It's a dual or double omics. It's a transcript and also it's the immune cells. But anyway, both of these cells are in both of these conditions, it's a transcript expression of the immune system and also the breast cancer cells. And from this omics, you get the hub genes, and then we will drug repurpose, and then discover new drug using this plant. And then from there, so basically this is her project lah. So this is her project. So overall, what she did is, we don't collect samples from patient. So what we do is we go to this database, Geo, and then we download the transcript data, transcriptomics data for metastatic breast cancer and also tolerogenic dendritic cells. So once we have these two, we will identify differentially expressed genes, DEGs, using GeoExplorer. And then from there, she will do common genes and whatnot lah. You can go through, and then she do enrichment analysis, and then protein-protein interaction. And then after that, this is where she finally identify the hub genes. And then the hub genes will now be tested for drug repurposing using several database of drugs. And then validation and verification. She will do bioinformatics for the ADMET toxicity. And then finally, she will do lab laboratories to verify that the hub genes are expressed in the breast cancer. And then when we treat the cells having the hub genes with the drug that we found from the in silico, from the analysis, she will test it whether it will change the phenotype of the cells. And in this study, the verification, she will use real-time PCR to validate the hub genes. So this is the example of how omics-based therapeutic study conducted at Inform under my supervision. Okay, so you identify two problems, you want to connect these two, and then you use all the tools, available tools, software, and database, and then you verify it in the wet laboratory. And then from here, you will show that the hub genes that you identify from this omics-based study can be used as a target for therapy. Okay? So basically, this is one example lah. I think that's the last slide. Yeah, that's the last slide from this chapter. So that's basically what chapter 1 is all about from my side. And Dr. Shikin will probably cover go back to the. So Dr. Shikin will share with you how omics-based therapeutic solution has been used in her group, because Dr. Shikin, she is based at pathology department, at medical school. And here in Health Campus USM, pathology department has NGS, Next Gene Sequencing Illumina Minisec. So they are now currently using that NGS for cancer purposes. So that's why we invite Dr. Shikin, because she was the person in charge for the machine, the NGS and pathology. And so we invite her